picture the human cell as this perfect impermeable fortress wall, right? Just a lipid bilayer guarding all the precious, highly regulated machinery inside.
Yeah, it looks incredibly secure from the outside.
It really does. But today, we are going to completely shatter that illusion for you.
Yeah.
Because we are looking at pathogens that don't just breach the fortress wall, they hack the gate's actual source code.
They absolutely do.
Welcome to this deep dive into the front lines of molecular and clinical pathogenesis. Our mission today is to unpack the precise, highly technical, tactical maneuvers that microbes use to establish a foothold, deploy chemical warfare, and ultimately survive the long game against both our immune systems and our drugs.
Right. And to do that, we're analyzing some incredibly detailed clinical texts and molecular pathway diagrams just to see exactly how this microscopic battlefield operates.
Because this isn't a basic biology review.
No, not at all. Setting aside the macro-level view of infections, the core of what we are dissecting today is molecular subversion. I mean, to truly master clinical infectious disease management, you have to understand the specific mechanisms a pathogen uses to manipulate host architecture.
Right, moving beyond just the basic clinical presentation of an infection.
Exactly. We are looking at the biochemical hijacking of our own kinases, our cytoskeletal networks, and our signaling cascades.
Okay, let's unpack this.
Yeah.
Because the physical barriers of the human body are obviously formidable. Before a patient ever presents with, fever or tachycardia or localized erythema, the pathogen has to secure an anchor.
And they have to do it in a fluid-swept environment, which brings us to adhesion.
Right.
It isn't just a passive stickiness, It is a highly specialized ligand-receptor interaction. The clinical diagrams we're looking at highlight uropathogenic E. coli, or UPEC, is a prime example here.
Because the urinary tract is a high shear stress environment. I mean, anything not firmly bolted down is just getting washed away.
Oh. Exactly. And UPEC overcomes this by deploying type 1 pila, which are tipped with this terminal adhesin protein called FIMH. And that FIMH adhesin binds with remarkable targeted affinity to highly manosylated proteins.
Specifically, the uroplakins on the surface of the host's uroepithelial cells.
Yes. It essentially creates a physical tether that resists the hydrodynamic forces of micturition, but that tether is really just the preamble.
Just the first step.
Right, because once they're anchored, they don't just sit on the epithelial surface, they initiate invasion. They actually force the host cell to undergo endocytosis, or they dismantle intercellular junctions to penetrate deeper into the submucosa.
And this is where it stops being a simple home invasion. I mean, the bacteria don't throw a brick through the window. It's like a hacker rewriting the smart home's operating system. They modulate phosphonocetide metabolism and erode GTpases to force the house to open its own doors and swallow the intruder whole.
The mechanism they use to execute that hack is breathtaking. Many gram-negative pathogens utilize a type 3 secretion system, or TTSS.
The molecular syringe.
Right. This complex needle-like appendage that injects bacterial effector proteins directly across the host cell's plasma membrane and straight into the cytoplasm. So if we look at Salmonella, it translocates several highly specialized effectors to orchestrate its entry.
Okay, walk us through that.
Well, first, it injects an effector called CYP-C, and CYP-C directly nucleates actin, which drives massive actin polymerization and the bundling of filaments right at the precise site of bacterial attachment.
Wow.
Yeah. And then it deploys a second effector, SOAPy, which acts as a guanine nucleotide exchange factor to activate the host's own Rho GTpases.
Specifically RAC1 and CDC42.
Yes, exactly.
And those activated Rho GTpases are what trigger the massive cytoskeletal rearrangements, creating those giant membrane ruffles that literally reach up and engulf the Salmonella bacterium.
They do. But honestly, the most elegant part of this subversion happens once the bacterium is safely inside the host cytoplasm.
What does it do then?
Salmonella injects a third effector, SPDP, and SPDP acts as a GTpase activating protein, doing the exact opposite of SOAPy.
Wait, really?
Yeah, it actively shuts down those same Rho GTpases, halting the actin polymerization and neatly closing the plasma membrane behind it. It orchestrates the breach and then meticulously cleans up the structural evidence.
So it opens the door, walks in, and shuts it using our own internal doorknobs. That is wild.
It is, but not every intracellular pathogen uses the TTSS syringe and membrane ruffling approach.
Right. The source material highlights Listeria monocytogenes as utilizing a completely different, far stealthier entry mechanism.
Exactly. Listeria relies on what is called the NLA-dependent pathway. It expresses a surface protein called internalin A, or INLA, which is anchored to the bacterial peptidoglycan by a transpeptidase enzyme called sortis.
And this NLA protein specifically targets E-cadherin, which is, a fundamental human cell adhesion molecule.
Yes, the one normally responsible for keeping our epithelial cells glued tightly together. By binding E-cadherin, Listeria subverts the host's adherens junction machinery. It hijacks beta-catenin and alpha-catenin.
And to generate the actual contractile force required to pull itself into the host cell, it co-opts the host's own motor protein, myosin VI.
Exactly. I want you to consider the complexity of that for a second. A pathogen carrying precise molecular keys to unlock our specific cellular adhesion molecules.
You really just have to pause and consider the sheer evolutionary complexity. So, okay, the pathogen has successfully breached the primary barrier. It's inside the host tissue. But the extracellular space in the submucosa is heavily patrolled.
Oh, very heavily.
You've got neutrophils, macrophages, the complement cascade. The heavy infantry of the immune system is actively hunting. So the invader needs a safe living arrangement to multiply.
Right, and clinical microbiology categorizes these residency strategies into 3 distinct groups. First, you have extracellular obligatory pathogens, like Streptococcus pneumonia.
The ones with the dense polysaccharide capsules.
Right, to fend off phagocytosis while they multiply in the extracellular space. Second, you have intracellular obligatory bacteria. These are the true squatters, like chlamydia and rickettsia.
Over evolutionary time, they've shed so much of their genome that they lack the metabolic pathways to synthesize their own ATP, or essential amino acids, right?
Exactly. They absolutely must live inside a host cell to survive. But the third category is the one that really challenges clinicians, the intracellular facultative bacteria.
These are pathogens like Mycobacterium tuberculosis or certain Salmonella serotypes.
Yes. Facultative intracellular bacteria possess all the biochemical machinery required to survive and replicate independently in the extracellular space. Yet during an infection, they actively seek out and invade host cells,
Frequently targeting the very professional phagocytes like the macrophages that are specifically designed to destroy them.
Right. And clinically, these infections are notorious for their slow evolution, granuloma formation, extended periods of clinical latency, and frequent reactivations years after the initial exposure.
Okay, wait, I have to push back there. If a facultative bacteria is perfectly capable of surviving in the extracellular fluid, why on earth would it deliberately choose to dive into a hostile environment like a macrophage?
It's a fair question.
You were talking about a cell packed with lysosomes, hydrolytic enzymes, oxidative burst machinery. It's literally diving into a vat of acid.
It seems counterintuitive, I know, but it's a brilliant evasion tactic. Because while the extracellular space might not have acid vats, it is heavily surveilled by circulating antibodies and complement proteins.
So they would rapidly flag the bacteria for systemic destruction.
Exactly. By forcing their way into a macrophage, they completely hide from humoral immunity. Of course, to survive the acid vat, they have to subvert the macrophage's intracellular killing mechanisms.
Right. Mycobacterium tuberculosis, for instance, secretes effector molecules that alter the host's Rab GTpases.
Yes, and normally those GTpases coordinate the fusion of the bacteria-containing phagosome with the destructive lysosome.
But by disabling them, TB arrests the maturation of the phagosome.
The lysosome never fuses. The bacteria effectively turn the macrophage into a mobile, heavily armored transport vehicle, completely shielded from systemic immune surveillance.
And that dictates a massive clinical takeaway for anyone prescribing therapeutics. Humoral immunity antibodies cannot cross the host cell membrane to reach these hidden colonies.
No, they can't. T cells have to take the lead. They can recognize infected macrophages and induce apoptosis.
And from a pharmacological standpoint, if you prescribe a powerful bactericidal antibiotic that looks great on a susceptibility panel, but it has poor lipid solubility, it's virtually useless.
Completely useless. The drug has to possess excellent intracellular penetration, like macrolides or fluoroquinolones, to actually reach therapeutic concentrations inside that host macrophage.
Because if it doesn't cross the lipid bilayer, the clinical infection simply persists.
Exactly. Now, just to provide some context, other microbes have entirely different residency strategies. Viruses are the ultimate obligate intracellular parasites, relying on profound immunomodulation and cytopathic effects.
And fungi often rely on tissue invasion, focusing on morphological switching to Pierce architectures.
Right, but bacteria excel at something else entirely. They are masters of biochemical warfare.
Here's where it gets really interesting. Because surviving in a stealthy bunker doesn't explain the massive multi-organ damage we see in the ICU. To do that kind of systemic damage from a hidden position, the pathogen has to deploy its chemical arsenal.
It has to shift from local evasion to systemic disruption. When analyzing bacterial toxins, clinical texts strictly divide them into endotoxins and exotoxins, and that distinction is critical.
Okay, let's break that down.
Endotoxins are strictly structural. They are a fundamental component of the bacterial architecture, specifically the lipopolysaccharide, or LPS, found in the outer membrane of gram-negative bacteria.
So LPS isn't actively secreted as a weapon.
No, it is released when the bacteria rapidly multiply, or when they are lyzed by the immune system, or even by beta-lactam antibiotics. The free lipid, a portion of the LPS, binds to toll-like receptor 4 on host macrophages, triggering a severe inflammatory cascade.
Exotoxins, on the other hand, are active, targeted munitions.
Yes. Exotoxins are highly specific, soluble proteins actively synthesized and secreted by the bacteria. These are the molecules responsible for the profound clinical presentations of diseases like tetanus, diphtheria, and botulism.
Right. Clostridium botulinum secretes an exotoxin that cleaves the snare proteins at the neuromuscular junction,
Completely blocking the release of acetylcholine and causing flaccid paralysis. And the critical clinical caveat here is that exotoxins are unaffected by antibiotics.
Wow, really?
Yes, you can administer broad-spectrum agents and eradicate every single vegetative cell of C. botulinum in the patient's body. But if that exotoxin is already circulating and internalizing into presynaptic terminals, antibiotics will do absolutely nothing to reverse the paralysis.
You have to neutralize the toxin itself using a specific antitoxin.
Exactly.
But beyond those classic exotoxins, there is a class of pathogens that deploy what are known as superantigens. If regular toxins are like conventional bombs targeting a specific cellular pathway, superantigens are, well, biological sabotage on a systemic, catastrophic scale.
Superantigens completely short-circuit the host's adaptive immune system. In a healthy immune response, an antigen-presenting cell ingests a pathogen, degrades it, and presents a tiny specific peptide fragment on its MHC class II molecule.
And this complex is presented to a T-cell receptor.
Right. It's a highly specific interaction that activates only the exact T-cell clones meant to fight that specific antigen, usually about 0.001 to 0.01% of the body's total T-cell population.
It's a precise, localized sniper strike. So how do superantigens bypass that regulation?
Well, superantigens like toxic shock syndrome, toxin 1 produced by Staphylococcus aureus, do not require intracellular processing. They bind directly to the outside of the MHC class II molecule on the antigen-presenting cell and cross-link it to the variable beta domain of the T-cell receptor.
They just act as a molecular clamp.
Exactly, forcibly locking the two cells together independent of any specific antigen recognition. Because they target conserved regions of the T-cell receptor, this unnatural cross-linking can activate up to 20% of the host's entire naive T-cell population simultaneously.
And the result of 20% of your T-cells firing all at once is the cytokine storm. You have a massive systemic surge of soluble messenger proteins molecules ranging from 6 to 70 kilodaltons, like IL-1, IL-2, and TNF-alpha.
Right. And those cytokines are intended for localized, precise immune coordination. When they are dumped into the systemic circulation in those sheer quantities, they become profoundly toxic.
Driving the exact pathophysiology of distributive shock and sepsis.
Exactly. TNF-alpha and IL-1 trigger massive nitric oxide production, causing widespread vasodilation. Simultaneously, they cause profound endothelial dysfunction. The tight junctions between the endothelial cells lining the capillaries just break down.
The structural integrity of the vascular bed simply falls apart.
Fluid leaks massively out of the vasculature and into the interstitial tissues. Intravascular volume plummets, causing severe hypotension. The cytokines also induce tissue factor expression on monocytes, triggering disseminated intravascular coagulation.
Microvascular clotting that chokes off blood supply to the organs.
Ultimately leading to refractory shock, ARDS, and multi-organ failure.
It is the ultimate, terrifying biological irony. The pathogen itself isn't directly destroying the host's kidneys or lungs. It is maliciously manipulating the host's own defense network into triggering a fatal systemic collapse. The immune system becomes the weapon of its own destruction.
And intervening early in that biochemical cascade is the primary challenge in critical care.
But let's assume the host survives. Let's say the patient is in the ICU. They receive aggressive fluid resuscitation, vasopressors, and targeted high-dose bactericidal antibiotics. the systemic attack is quelled.
The bacteria are losing the acute battle.
Right. But they don't just die. They go into stealth mode to survive the long game, because they have evolutionary contingencies for this exact scenario of overwhelming stress.
Exactly. If they can't win the acute battle, they dig trenches. And the most prominent strategy here is the formation of biofilms.
Which aren't just disorganized layers of slime on a catheter.
No, they are highly structured three-dimensional bacterial cities. The clinical texts outline a very specific progression here. It begins in stage 1, where individual free-floating planktonic bacteria make an initial physical adhesion to a surface.
This could be native host tissue, like a heart valve, or abiotic surfaces, like an orthopedic implant.
Great. Once secured, they move into stage 2, utilizing specific ligand receptor complexes and poly to aggregate and form early microcolonies.
And then they mature into a massive 3D community, completely encased in a protective envelope made of peptidoglycan, proteins, and exopolysaccharides.
And that extracellular polymeric substance acts as a profound physical and chemical shield. It functions as a diffusion barrier.
Right.
As antibiotics attempt to penetrate the biofilm, they bind to components in the matrix or get degraded.
Wow.
Furthermore, the microenvironment inside the mature biofilm is highly altered. The bacteria at the deep core experience severe hypoxia and acidic pH, which significantly lowers their metabolic rate.
And since many bactericidal drugs like beta-lactams require actively dividing cells to be lethal,
The dormant bacteria at the core of the biofilm remain completely untouched, ensuring the infection's persistence.
but what about bacteria that don't form these massive structured biofilms? How do individual bacteria survive heavy, sustained antimicrobial bombardment inside host tissues?
That introduces a phenomenal cellular adaptation called small colony variants, or SCVs.
Yeah.
The molecular diagrams detail this using Staphylococcus aureus as the primary model.
Okay, how does that work?
Well, when a standard wild-type colony of S. aureus is subjected to severe, prolonged environmental stress, which could be hypoxia, nutrient starvation, the hostile intracellular environment of a macrophage, or exposure to sublethal antimicrobials, they undergo a radical phenotypic switch.
It's like a rogue submarine going on silent running. To avoid detection by depth charges, which are our antimicrobials, the SCV submarine completely cuts its engines, dropping its metabolic activity.
That's a perfect analogy. The metabolic downregulation is staggering. The bacteria drastically reduce their electron transport chain activity and downregulate the tricarboxylic acid cycle. Their metabolic rate slows to a crawl.
And because their metabolism is virtually halted, their cell wall synthesis stops, right? And because beta-lactam antibiotics rely entirely on disrupting active cell wall synthesis to cause lysis, the SCVs become essentially invisible to the drugs.
Their antibiotic resistance skyrockets, but, and this is key, not through the acquisition of a new resistance plasmid or a genetic mutation. It is entirely through sheer metabolic dormancy.
That is so frustrating clinically.
It is. In a lab, these SCVs form tiny, atypical, pinpoint colonies on an agar plate that take days longer to grow, meaning they are frequently missed or dismissed as normal flora.
So they establish long-term persistence in the host tissues, surviving in a suspended state.
And then weeks, months, or even years later, when the antimicrobial pressure is finally removed and host conditions improve, they undergo a reverse phenotypic switch. They upregulate their metabolism, revert to their aggressive wild-type state, and the patient presents with a sudden, inexplicable clinical relapse.
The tactical brilliance we've unpacked today is staggering. I mean, we started with the physical breach, looking at pathogens, utilizing targeted phage adhesins and type 3 secretion systems to subvert host row GTpases, manipulating our own actin cytoskeleton to force their entry.
Yeah.
We explored how facultative intracellular bacteria deliberately dive into macrophages, altering Rab GTpases to halt lysosomal fusion. We broke down how superantigens trigger a devastating cytokine storm. And finally, we saw how bacteria retreat into impenetrable biofilms or trigger a phenotypic switch to become dormant SCVs.
The central theme uniting all these mechanisms is that human pathogens do not merely overpower the host with sheer numbers.
No.
They outmaneuver us through precise molecular subversion. They possess a terrifyingly intimate knowledge of our cellular machinery.
So what does this all mean for the clinician staring down a persistent, multi-drug-resistant infection?
Well, it forces us to reconsider the entire future of infectious disease management. For a century, the paradigm has been bactericidal designing molecules that poison the invader's machinery. But given that bacteria rely on hijacking our own cellular machinery and respond to antibiotics by hiding as SCVs, that paradigm is failing.
So what's the alternative?
The emerging frontier is host-directed therapies. Instead of targeting the rapidly mutating bacteria, we target the host. We use pharmacological agents to temporarily lock down our own cellular doors.
Oh, wow.
Yeah, if we can modulate our own kinases, temporarily inhibit our own Rho GTpases, or block the specific intracellular fusion events that facultative bacteria rely on, we could render the host completely inhospitable, effectively starving the pathogen regardless of its antibiotic resistance profile.
But if we start using host-directed therapies to systematically lock down our own cells and alter our own cellular pathways, what happens to the beneficial commensal microbiome that likely relies on those exact same pathways to communicate with our immune system? Are we going to accidentally lock out our cellular allies and trigger widespread autoimmune consequences while just trying to starve our enemies?
It is definitely a complex, double-edged sword.
Something to really think about. Well, we want to thank you for joining us on this deep dive into the microscopic battlefield. We hope this highly technical exploration of pathogenesis has armed you with a new perspective on the molecular wars raging just beneath the skin. Keep pushing the boundaries of your knowledge, and as always, keep questioning the clinical dogmas. We'll see you next time.